So good afternoon, everyone. On behalf of myself, Wendy Spearman, and my colleague, Mark Sondra, we welcome you to our monthly COVID-19 Echo webinars. We've got a very exciting lineup today, looking at the local epidemiology update, but importantly also looking at our understanding of the variants, particularly the Delta variants. And our chair for today is Graham Mankes. Over to you, Graham.
Right. Thanks very much, Wendy, and welcome to everybody who is attending this webinar. It's our monthly ECHO Department of Medicine COVID-19 webinar, and we really find ourselves in the midst of a very severe third wave of COVID in the Western Cape, with substantial numbers of patients requiring respiratory support in our hospitals currently. And we know that this is driven by the Delta variant in a large way that was first recognized in South Africa just 3 months ago, but has come to dominate the epidemic that we've seen currently and the third wave. So we really have talks today that speak and are particularly relevant to our current situation. In the program that we circulated, we had 2 speakers, but we're actually going to have 3 speakers today that are going to deal with the issues around the epidemiology of what we're seeing in the Western Cape currently, and then understanding of the variants with obviously a specific emphasis on Delta. And then finally, look at a local study that's trying to understand the implications of the Delta variant on our epidemic in the Western Cape. So our speakers today, and I'll introduce all 3 speakers, and they will go through their talks one after the other, and we'll have a question and answer session at the end. First speaker is Marianne Davies. Marianne is a public health specialist at the UCT School of Public Health, as well as the Western Cape Department of Health, where she is responsible for epidemiology and surveillance. Marianne will be followed by Marvin Hsaio. Marvin is the acting head of the Division of Medical Virology at UCT. And Marvin will then be followed by Hannah Hsaio, who is a registrar in public health at UCT, as well as currently working at the Western Cape Department of Health Data Center. So over to Marianne, and as I say, we will have the 3 talks and then time for question and answers at the end, and please type any questions you have in the chat as you go, and we'll address them hopefully at the end. Thanks, Marianne.
Great, thanks very much for the introduction, and once again for the opportunity to join the ECHO series. Can I just confirm that you can see my screen and hear me?
No, Marianne, we can't pause there.
You can't see my screen. Okay, let's try again.
Share again, please.
Is that better?
There we are.
Okay, perfect. So this is where we find ourselves in the Western Cape at the moment. As Graham has said, we are in a severe third wave, which is looking to approach what we saw in the second wave, although not quite reaching those levels yet. But we have seen some very high case numbers over the last few weeks, and I'd like to just focus in on that a little bit. And last week in particular, we were noted to have our single highest number of daily cases, and the Twitter famed Ridwan Solomon, who regularly tweets on epidemiology, noted that we'd reached a new daily record. What I would like to highlight is that in this wave, compared to in the previous waves, we've seen quite a bit of lumpiness in the case data. And so compared to the steep and smooth increases in that gray dotted line, which is the 7-day moving average of cases, in this wave, we've seen this kind of more lumpy picture. And there are a couple of reasons for that, which I'm going to go through in the next slides. So, sorry, wrong direction. So here we're looking at our usual provincial resurgence overview slides with the cases at the top. And what you can see is that in sort of mid-July, we had this plateau in cases. It looked like we'd reached the top of Table Mountain, and we all felt relieved, although I must say some of us felt this looks too good to be true. The contributions to that plateau were probably largely related to the taxi violence and people not accessing testing. And then just after that, we saw this huge steep increase in cases, our record number, which was on Monday last week. So that was probably partly return to testing, and people were now too sick to avoid testing. But also what we have found is that with the expansion of antigen testing, that data tends to come in a much more lumpy way because it has to be manually captured. It's not coming in an automated way from the lab. And so there are often delays, and it's not always clear whether the date on which the case is reported is actually the day that the test was done. And so, for example, that particular high peak was because we received about 1,000 antigen tests from one lab reported as being done on that day. And most likely, those were tests distributed over the previous 2 weeks, but we have no way of knowing what day those cases were actually diagnosed. In terms of the other metrics on the bottom left, we've had a steady increase in proportion positive up to about 40%, an increase in admissions, although you can see even in the admissions, there was a bit of a plateau related to taxi violence, suggesting that even people who were very sick couldn't get to hospitals, but now reaching about 350 a day. And then in terms of deaths, we're now just over 100 deaths a day. In comparison, in our second wave, we were in excess of 150 deaths a day at the peak. There's also been some concern because we had a couple of days where no new deaths were reported last week. So just to explain for those who do follow our data very carefully, the date of reporting is not the same as the date of death, and there was some curation of deaths that happened last week, which resulted in no deaths being reported on a particular day. But if you actually do look at the date of death, you can see the steady increase, and there's nothing too strange going on there. Looking at the excess deaths, we're now in our 6th week of steep increases and exceeding the upper bound of expected. And we're hoping, because last time it took 6 weeks to turn, that this might mean that we are heading towards the turn. And then finally, the reproductive number. This is really just calculated based on the cases and deaths. And so when we do get this lumpy case data, it does cause this kind of bumpiness in the reproductive number, but still sitting currently at about 1.1. This is just looking very quickly across the metro, and you can see this pattern of the plateau followed by a steep increase is present in all sub-districts in the metro, perhaps a little bit less in Khayelitsha. And the wording at the top shows that we've also seen this shift. If you look at the bars towards the left-hand side of all of these curves, it's mostly brown bars, which indicate private sector testing. And on the right-hand side, we've seen a shift to more dominance in the public sector. And I'll talk to that a little bit later. In rural, again, still seeing increases in all sub-districts, particularly Central Karoo, but of course Central Karoo is small and with small numbers overall. Because of this lumpiness in the case data, it's meant that doing any kind of forecasting is very difficult. And so the South African COVID Modelling Consortium does do short-term forecasts. I call them the weather forecast. And what you can see on this graph, the black line is the previous cases. The gray line is the forecast for the current week, and then the actual cases. And you can see that over the last few days, the actual cases exceeded the forecast. And that means that when they forecast for the next week, that has pushed up the forecast to over 5,000 cases. But because of what I've explained around having an excessive number of cases last week, that probably is an overestimate, and we certainly hope that we don't see 5,300 cases on the 11th of August. This also means that the prediction around whether we've passed the peak has moved around a lot. It was a couple of weeks ago in the plateau, they said there was about a 75% chance that we'd pass the peak. That went all the way down to 9% last week, and fortunately is looking a bit better at 27%. But really just highlighting that the case data is difficult to get a sense of where we are, but certainly admissions and deaths still increasing. I'd like to spend a little bit of time comparing the 3 waves and what can we learn from that. So what we're going to look at here is the first wave in the light blue, the second wave in the dark blue, and the third wave in orange. And you can see that across the province, in terms of cases, the third wave is now approaching the peak of the second wave with those very high case numbers. But there's a little bit more to the story than that. So what this graph is showing is on the left-hand side cases, in the middle admissions, and on the right-hand side deaths. And the top graphs are for the public sector and the bottom for the private sector. And there are a couple of things to point out. The first is that both in public and in private, the case numbers, although those are reaching quite high numbers, admissions and deaths still haven't quite reached our second wave levels. And then what we see very clearly in these graphs is how the wave took off in private first. And there was this delay in the public sector, and the public sector cases really only increased quite a bit later. And although we've seen a plateau in private, in all the metrics, in the public sector, we are still seeing increases. We know that this wave is being driven by the Delta variant, and Marvin's going to talk a lot more about variants shortly. And we know that that number of cases very much correlates with the proportion of all specimens that are due to Delta. And this is a slide produced by the Network for Genomic Surveillance and the NICD. And what we also know is if you look at those early cases of Delta in weeks sort of 18 to 22, when it first seemed to be emerging in the province, is that it emerged first in the private sector. And in fact, we were fortunate that we were doing some private sector surveillance, and we expanded it at that stage because we could see that something different was going on in the private sector. And so that matches with what we've seen in the epidemic. And it also seemed to emerge first in the garden route. And in fact, our first public sector Delta specimen was on the 24th of May from the garden route. And you'll see how that is relevant in the next couple of slides as well. So again, this pattern of comparison across the waves is a little bit different for different areas. Here we're looking at different sub-districts. And this is again looking at cases. And you can see that in the metro, for most sub-districts, we're still a little bit off the second wave peak, but Klipfontein, Mitchells Plain, Northern and Tygerberg are both at the same level as the second wave. And in rural Central Karoo, Garden Route and West Coast have either reached the same level or exceeded their second wave peak. Admissions and deaths are kind of following this pattern. And what I want to highlight in these rural areas of Central Karoo, West Coast and Garden Route is that we are actually seeing pretty much the same or even higher deaths than we saw in the second wave. And definitely in Garden Route, we're seeing more deaths at a peak, although that was it's a steeper peak than we had in the second wave than we had in the second wave. Now, the question about seeing these more deaths is this because Delta might be more severe and causing more severe disease? And we know that there is some published data to suggest that Delta may be associated with more severe disease. We haven't had comparisons necessarily with Beta, but in Canada, Singapore and Scotland, they've shown higher risks of more severe disease in people with Delta compared to other strains. So the question is, are we seeing the same in our setting? One way to try to look at this is to compare across the waves in terms of the outcomes that we're seeing. And for all of the graphs I'm going to show, just to highlight, this is limited to the Cape Town Metro. The reason for that is because it's easier to define the wave periods for just one particular district. It's limited to the first part of the wave because what happens in the second part of the wave is quite different to the first part, and we haven't seen the second part of wave 3 yet. And it's limited to known public sector adult patients, so who we know about their comorbidities. And importantly, it's limited to people where their first diagnosed infection is in the respective wave, because we don't want to include people where this may be a reinfection. Acknowledging that diagnosed infection is, of course, a complete underrepresentation of all infections. So one of the things that we found in each of these waves is that people have tended to say as the surges happen, that they're seeing younger and sicker patients. And so the top graph on the left-hand side is showing the proportion admitted and deceased in different age groups across the waves. And what you can see is that that's actually not the case at all when we actually look at the data, that we've seen fewer younger people in subsequent waves, and if anything, more older people, particularly in the over 70s. If we look at the probability of death by 28 days since diagnosis by age group and wave 1's on the left and wave 3 is on the right for each of these, you can see that there's really not much difference. There's some hint in the over 70s that we might be seeing some increased probability of death in wave 3, but the confidence intervals are quite wide and do still overlap. And then doing an adjusted analysis to look at the adjusted hazards of death by wave, and this is adjusted for age, sex, all known comorbidities, and for admission pressure and subdistrict. Cases are at the top and admissions are at the bottom, and I've used wave 2 as the reference. You can see that for cases, wave 2 was worse than wave 1. Wave 1 is protective, and that wave 3, we're seeing about a 30% increased risk of mortality. And among admissions, we don't see a difference between wave 1 and wave 2, but wave 3, we do see an increased risk of mortality. These wave comparisons are very flawed because we know that things are very different across the waves in terms of testing criteria and treatment protocols. And so what we really need is to know who has and hasn't got a different strain of the virus, and look at their outcomes in the same wave during the crossover period when the new variant is taking over. And the presentation by Hannah is going to start to try to get at that. And the second thing that's a challenge about these wave comparisons is just to note that we are only halfway through this wave. And what we did find in the last wave is that things actually changed. And by the end of the wave, we did see increased risk of death in both cases and admissions in wave 2 compared to wave 1. The other thing, if we look at comparisons, is not just by subdistrict, but actually if we go down to particular individual suburbs in different low income areas, is the different impact of wave 3 in places that had a very big wave 1 compared to places that didn't. So at the top, we're looking at places where they had a sort of moderate sized wave 1 and a similar sized wave 2. And you can see that wave 3 looks pretty much the same, even in these low income communities. At the bottom are places like Khayelitsha, which I think everyone's very familiar with, which had a much bigger wave 1 than wave 2, and we've got some other similar communities. And in all of those, we're not seeing much in terms of wave 3, and really a very dampened down wave 3. And so that kind of suggests that communities with high seroprevalence and where they have had many prior infections do seem to have some protection against subsequent waves. So we do know from Cheryl Cohen's first study, which was presented at the last ECHO, that prior infection in our setting does seem to be protective against subsequent infection. And I just wanted to have a quick look at whether this is playing out at the level of communities. And so what we're looking at here is the difference in the hazards of death by wave in Khayelitsha, where we know that seroprevalence was high and where we know that there was this very severe first wave. And you can see that in contrast to the differences that we're seeing across the metro as a whole, there really was not much difference between waves in Khayelitsha, but those confidence intervals are very wide. The other way I looked at this was to use HIV status as an indicator of living in a community with high levels of prior SARS-CoV-2 infections. And the reason for taking this approach is because we do know that seroprevalence in people with HIV has been higher, even in communities where the overall seroprevalence was a bit lower. And so we saw, for example, at the end of the second wave, that people with HIV had seroprevalence around 60% compared to diabetics, where it was around 47%, even in the public sector. And what we have seen is that people with HIV has seemed to be somewhat protected in subsequent waves. You can see the gray in wave 3, really much lower proportion of people with HIV among the cases, admissions, and deaths compared to earlier waves. And on the right-hand side, doing the same wave comparison at the top in people with HIV, not because I think HIV has any biological protection, but just because I think it's a proxy for living in a community where you couldn't socially distance in wave 1 and you were very likely to have had COVID. And then at the bottom, people without HIV, you can see again that we don't see any difference by wave in the people with HIV, but we do see the effects that we've seen across the metro as a whole, with wave 2 looking worse than wave 1 and wave 3 looking worse than wave 2. So finally, Graham asked me on Monday if there was any impact. Are we seeing any benefit from vaccines yet? And we now have about 60% of our population over 60 who have received a vaccine. And we do hope in a future ECHO webinar to do a much more thorough presentation looking both at the impact of reinfections as well as vaccines. So this is just a really crude look. So if we look before the third wave really got going, you can see the number of vaccines that were given. And we had a relatively small number of people who, by the beginning of July, had received at least 1 dose of their Pfizer vaccine and where 2 weeks had elapsed. So we might expect some level of effectiveness. And so I don't want anyone to construe this as an effectiveness analysis, and the data is really still needs to be interrogated. But what I want to highlight is that in comparison to the 35% of the population who had been vaccinated, we see things going in the right direction in terms of relatively smaller proportions who've had that 2 weeks since dose 1 among the cases, admissions, and deaths, which is reassuring. I think there are many caveats to looking at this data. Most importantly, we don't expect that much protection just from a single dose of Pfizer. Many of the people who would have been vaccinated early might have been admitted people or people living in congregate long-term admission facilities, and so might be quite elderly and have a high risk of death anyway. And of course, we're not taking into account any kind of adjustment for the fact that those who accessed the vaccine early might be very different in a number of ways from those who don't. So watch the space for a more robust analysis in weeks to come. And with that, I'll hand over to Marvin to explain to us a little bit more about why Delta is causing such a headache.
Thanks very much, Marianne. So we'll go over to Marvin for the discussion of the variants.
Thanks, Graham, and thanks, Marianne. So Graham gave me this topic, understanding variants, but I adopted, but in hindsight, I think this is a bit of a grandiose topic, and you know, I'm acting as the kind of palate cleanser in between the two speaker that has a very kind of data and analytic approach, and I think my middle bit is more of a kind of a story around, you know, how the various different variant came about and where we are going. So at best, this is a kind of oversimplification of a very complex system, and at worst, like many things, I could be completely wrong. But nonetheless, we can't talk about a variant without a kind of understanding kind of evolution in general. So I'm just going to quickly talk about evolution, particularly point mutation, which is primarily what we are talking about in terms of coronavirus evolution. And looking at this graph on the left-hand side, This is a paper from Science more than 10 years ago. And basically what you can see here is that the point mutation rate is inversely proportional to size of genome of the organism. And if one compared the different various viruses, the smaller RNA viruses mutate at a fastest rate, whereas the DNA virus are doing so at a much slower rate. And coronaviruses, they have among the largest genome, at least the linear RNA genomes, about 30,000 bases. And they sit around here, which is amongst the kind of slowest evolving viruses. They have a mutation rate of only about 10 to the -6 per site per cycle of replication. And that is thanks to one of the non-structural proteins, NSP14, which has an exoribonuclease function. And basically, as this viral polymerase copies its genome in their reproductive cycle, this NSP14 has some sort of proofreading function to make sure that it doesn't kind of make too many errors, and the less errors the replication process makes, the less the likely is to mutate. And so what does this kind of 10 to the -6 per cycle mean in terms of kind of human pandemic time? The answer, you may have guessed, it depends how quickly it's replicating. And if we can put it just kind of in the pandemic terms. So this is a kind of cluster phylogenetic tree taken from NextStrain, who kind of gets data from GISAID, which South Africa and our sequencing network contribute all the SARS-CoV-2 sequence data. And just to orientate you, this is a one format of the tree where on the horizontal axis is the time, and the vertical axis is the number of mutations, the different sequence that's kind of moving away from the ancestral virus. And what you would see here is, as predicted, in a fairly linear fashion, as time passes, there are more and more mutation being accumulated. So the first thing to observe is obviously that there hasn't been any recent increase in speed of evolution. Is still maintained at the same speed. And I would just mention that actually, when we talk about variant and viral evolution, the most important evolutionary step actually happened way beyond here. So this is the mutation the virus has acquired in order for it to infect human being, binding to the ACE2 receptor and able to kind of replicate in any form of capacity. And because we don't know how long it took for this to happen, we may be looking at a very well adapted virus. So it depends where I believe their evolution may happen in the test tube, or more likely from another animal species. But another interesting thing to note is that if you look at the trajectory, at least the early divergence of some of these sequences, we're not actually seeing all these sequences completely fan out. And in fact, they're still clustering fairly along this kind of central line. And what this means is that as the virus spread in human population, there is a strong purifying selection. So the virus definitely has some constraint. It cannot just mutate to in any direction it likes. And I think that in part is the good news. And in fact, for the most of the first year of circulation, we hardly saw any kind of major mutations, and that has been sustained. And to put these variants in a slightly different tree. Now this, where you kind of swing the tree around to have the number of mutation on the horizontal axis. And some of you may often hear these variant of concern or variant interest that talk about these viral sequences that sit on a long branch length. And basically what that means is that compared to the other viruses that we have seen before, where in the previous slide where we see these kind of strong cluster together with very few mutations, these viruses have a high number of mutation that is accumulated compared to their ancestral colleagues. And what does this mean, and how does this how these kind of huge number of mutation accumulated all of a sudden? So this is a diagram taken from Katrina Lithgow's paper in Science in April this year. And it gives a really good idea of kind of various different mechanisms or the process by which viral evolution can result in kind of variants occurring in population. So in the beginning, we are infected by a relatively homogeneous viral population. And within a person, some minority variant can develop within that individual. But what gets transmitted on usually is the predominant variant, and that goes on to be the fitter virus to actually infect someone else, and the cycle repeats itself. But occasionally we can have some of these minor variants transmitted to another person, and depends on which of these variants are fittest. You have some of the variant emerging in another person. And finally, there is also the possibility that you can have a mixed infection being transmitted. So this is talking about the bottleneck of the infection and becomes wider, and then more than one of the variant can infect another person, or some sort of a co-infection situation can occur. So to get back to the question that I posed in the previous slides, how does new SARS-CoV-2 variant accumulate these high number of mutations? It is not because they're all arisen all of a sudden at the same time. It is there is a long evolutionary process that we haven't been we're not able to see, and then the end result of after we sampling, we're only finding this one that's variant that emerged, and it's because of evolution by three separate things. One is that there's a large scale community transmission, so a lot of these are being driven, so the fitter virus kind of survives and the new variants emerges. It could also be that you can have one person that can have a prolonged infection or have a chronic infection where this viral evolution can happen in one single individual. And this has certainly been documented in both people undergoing immunosuppressive therapy and people with severe HIV. And finally, you can have a situation where there is a transmission or co-infection, and this I think currently is under study, and we don't really understand this very well. So just to kind of go back, in terms of viral evolution, there's really two components. One is the virus, and the other one is the hosts. And from the viral component, that for variant to emerge, viral needs to replicate, and this takes time. And this mutation process is really random, and I can't iterate this, that the reason why these things are so difficult to predict and to understand is that this is essentially a stochastic process. So we there's an element of chance so that the virus can emerge at the right place at the right time, and obviously the more infection that takes place, the less time is needed for a beneficial set of mutations to emerge. And then one of the examples that very early on, and this is sometime in 2020, early on, there is a mutation that improved receptor binding and these 614G that emerged, and this mutation then it's become stuck in all of the kind of subsequent virus, almost the entire, the various different lineage here contain this particular mutation. From the host side, the reason why virus change is because there are selection pressure. So in other words, in order to drive this kind of fittest survival, the change in selection pressure, which is mainly host immune response. I mean, I try to come up with some other possible selection pressure, but really I think I'm clutching straws. It's mainly just host immune response. And in terms of host immune response, the primary selection pressure, and this is from the transmission standpoint, is actually neutralizing antibody that's exerting the selection pressure. And I will talk about this, in a bit. But I just, want to actually go back to more kind of epi-size to talk about, some of the phenomena that we're seeing. So the SARS-CoV-2 has been described as epidemic that's driven by super spreader events. And, this is kind of an opinion piece that's written by Muge Chavik and I think Stephen Burrell. I think this is also in Science. Basically, they refer to like, there are different transmission network patterns that results in different downstream number of cases. But you can see that, there's certain people either in the workplace or social engagement that are able to be in contact in a lot of more people. When they are infected, they are able to act as super spreader, whereas the other individuals aren't. And what this means is that, there are kind of number of kind of modeling study that estimated, I think this is some from the public health England contact tracing work. They find that, kind of 90% of the community transmission can actually be traced back to something like 2 to 5% of the infected individuals. And this kind of super spreading pattern can really kind of affect the variant and how they spread across the world. So even the world is kind of fairly connected, and much less so in the last 2 years. That, when you have one virus that's introduced in particular community where the level of immunity is pretty low, you have this what we call a founder's effect, and that is the first virus that's getting into a kind of virgin population completely unexposed, they are able to establish themselves as a dominant variant, even though compared to the other variant across the world, it may not be the fastest virus. And this is why that we see, for example, in South Africa, Beta variant has kind of emerged in sub-Saharan Africa. That's one of the dominant variant versus in Brazil, South America, where you see more Gamma, and in Europe and probably the rest of the world, it was Alpha. But the main change now is that across the world, what we are seeing as the dominant variant is Delta. And this, I think Marianne already showed you, very rapidly, and similar to how Beta has emerged in the previous wave, is that very rapidly from its first introduction, it became the completely dominant variant, where currently, I'll be very surprised when we see a sample that is not Delta variant. And this, to me, is not founder's effect, because at the beginning of the second wave, there are multiple different variants circulating, including what we thought the previous fittest virus, which was Beta. And this was quickly replaced by Delta, showing that Delta does have a survival advantage over its peers. So what has changed during all this time? And we're looking at this kind of global picture. And that has really been the kind of host immunity as a driver of selection pressure. In the first wave, we saw little or no immune selection, so the virus is just kind of freely replicating and doesn't need to make much adaptation. In the second wave, we're starting to see a fair amount of people previously exposed, but there's still a lot of people unexposed. So it's just a matter of kind of able to infect more individuals and getting into those populations. But when we get into 2011, and especially now, we start to see some highly immune population, and back to this whole super spreader driving the epidemic concept. To get into new communities and to get into the same communities where not everyone has been infected, one still rely on those kind of central node of those. We still rely on those central spreaders in order to infect other individuals. And this is where able to reinfect an individual or having mutation that confers some sort of immune escape has become critical. And I think this is the difference mainly that we see in the second wave, and especially in this wave, is that we have seen variant that has a lot of immune evasion potential. And this is, what we are seeing in terms of mutation profile. And I know most people, if you're like me, we don't really like these alphanumeric characters that resemble our Wi-Fi passwords. I'm not going to go into each of them, but there are kind of group of mutation, particularly around receptor binding domain that's associated immune evasion, and as well as some that's at the N-terminal domain. But the interesting new one in Virgin Delta is this particular one in P681R, and this happens near this S1, S2 furin cleavage site. And in my opinion, I think this is still a form of immune evasion, despite, a lot of people are calling this kind of either a virulence factor or a factor that kind of improve its transmission. And so there are more than one way of evading the immune system. You can either change the conformations, at the surface where the antibody binds, or I mean, in this approach, and you can see that the lion's test is on my mind. So like the way I can think of to describe this is called attack the gain line. And this is kind of concept that's explained very well by David always tweet a few days ago. And this is talking about, in people who have been exposed, and to a certain extent people have been vaccinated, we develop antibody, but then this antibody don't persist forever. We have B cells that have memories, and when we re-encounter the infection and the antigen, these B cells then expands and then provide a neutralizing antibody that protects us from severe disease. But if the virus can have a very strong, or much stronger than the ancestral variant, and replication in the beginning, in the first few days of infection, before the amnestic B cell response, these memory waking up, is able to produce enough antibodies, then it can overcome the infection and so-called breakthrough the infection. And in the end, the people probably still be protected because we have these B cells and probably T cell immune response, but then there is sufficient infection not only to establish in this individual, but potentially have onward transmission. And some of this concept, there's one paper in Ali et al. Looking at the cohort in Hong Kong compared to the 2021 epidemic versus the 2020 epidemic. So the latest one obviously is caused by Delta. That one sees that the viral load or ability to detect the virus very early on after infection is much higher than compared to the previous wave, and the viral load is also much higher. And we see this in our kind of local data as well. And this work is kind of confirmed by kind of different approaches. So this is coming from a Ravi Gupta's group, where they use a 3D organoid model to look at what is happening, when they establish infection, and they can see that the proportion of the spike that's in the cleaved form is much higher, in Delta compared to Alpha or the wild type, meaning that the spike from Delta viruses kind of have probably a advantage in terms of kind of a higher affinity, or are highly accessible to more different type of cells readily in the upper respiratory tract. So yeah, I probably rambled on a little bit, and with a lot of kind of information thrown at you. What does this all mean? I think, to kind of put it simply, and this is kind of the way that I try to kind of explain to my own mother, is that the virus had been and will always continue to evolve, and its goal is to able to infect more people consistently, despite the host immune response. And from what I've seen, there's really beyond what we are currently doing, the current MPIs, there are really little kind of public health interventions that we can do to stop this infection, and these variants are just besting us currently. And our only solution is, because we can't stop the infection, the only thing is that we can stop disease, and that's where the vaccine comes in. This is really our only solution, is to have vaccine that protect against disease. And only when we have all the vulnerable people, and probably not so vulnerable people, are protected from disease by the vaccine, then we can go back to our normal lives. So I'm going to end here, and I'd just like to acknowledge firstly the NGSSA network, which constantly provides interesting data and information, and I learned from all the different colleagues in that network tremendously, and also other people in the division who I regularly kind of talk to and bounce idea with, and really help my own solidify some of my understanding. So yeah, I thank you for your attention, and but I think I'm not going to stop talking here, because I'm just going to make a few introductions before Hannah takes over. And so we want to look at the impact of Delta, in our cohort in Western Cape. And the starting point of this analysis is that we started to notice around the beginning of June that when we're looking at one of our diagnostic tests, this is C-gene assay, which has 3 different viral targets. One particular target is starting to drift upwards. So CT value of this one is becoming increasingly high. So here you will see the plot here of a relative CT value of RDRP compared to E-gene. And you can see that we sensed that something that was going on, but at that stage we didn't know it was Delta virus. But what's happening is that over time, this has really become taken over a dominant effect. You can see here by July, 80 to 90% of our C-gene sample had this particular phenomenon. And the scientist in our division, Dr. Zia Valley Omar, he looked up all the sequence in the region where the primer binds, and he noticed that there is a overwhelming kind of predominant mutation where the primer binds among the South African Delta variant and also the Delta variant elsewhere. And this RDRP mutation could possibly cause the phenomenon in the RDRP dropout that we have seen. And so we looked at all the sequences that has kind of C-gene CT values, and you can clearly see here the relative RDRP CT is much higher to our previous variant, which is beta, but also alpha and the other kind of variant that's circulating at the time. So we thought this actually can be used as the Delta flag to look at, the kinetic of which the virus is kind of introduced into the population, and this could be a basis, in collaboration with our provincial colleagues to try and look at the impact on admissions, severe disease, death, in a very broad level, in a very high level. And on that note, I'm going to hand over to Hannah, and then she can kind of talk through the analyses that she has done.
Thanks so much, Marvin. Can you all hear me all right?
Yes.
Great. So I'm going to be discussing the clinical outcomes for this Delta flag that Marvin has introduced in the Western Cape public sector during the 3rd wave. And it's a survival analysis using routine observational data. So as Marvin has described, this Delta flag of partial RDRP dropout started increasing rapidly in the province around May, first in the Garden Route and then in Cape Town Metro, which was consistent with the findings from sequencing about how the Delta variant itself moved and rapidly overtook in the province. Next slide. But before we go into the actual analysis, we just need to talk a little bit more about the C-gene assay. So this is the assay that's used to determine this Delta flag, and it's not used uniformly across the province. When we looked at all the COVID-19 positive tests in the Western Cape, and we grouped it into the C-gene PCR, other PCRs, and antigen tests, you can see that the blue bar graph, the C-gene tests, are more commonly happening in the Garden Route and in the Cape Town Metro. And even within Cape Town, there's a lot of variation across the sub-districts. And you can see that the C-gene is used more commonly in the sub-districts that the first year NHLS lab services. Next slide. And so with this geographical variation in mind, we were a little bit concerned that the patients themselves might differ depending on the test type. And when we just did a sort of a quick look to see if there were any differences in age, sex, and comorbidity, there wasn't really much to be found. And there were quite a few with missing data, but those were mostly from the private sector, and they were excluded from the analysis. So next slide. So moving on to the actual survival analysis itself. As I said, we looked at all those who had a COVID-19 episode diagnosed from a C-gene assay from April 2021, and we restricted to only those in the public sector where we had the CT values to calculate this Delta flag and the comorbidity data needed for adjustments. We used Cox regression and looked from time of diagnosis to time of outcome. And the outcome was primarily admission with out-of-hospital deaths, as well as deaths alone. And the data set ended last week on the 28th of July. In all the analyses, we adjusted for sub-district of residence as well as hospital pressure. And by that, I mean the weekly public sector admissions in the province as a whole, as this allowed us to get an indication of the differences in the health system at the start of the peak compared to later in the peak, and so that we could adjust for this at least partially. Next slide. So getting right into the results, when we look at the severe outcome of admission, we see that the Delta flag was significantly associated with it. The adjusted hazard ratio was 1.43, even after adjusting for these various factors such as hospital admission pressure and geographical subdistrict. The usual risk factors for severe disease were also found, so male sex, increasing age, and comorbidities, particularly diabetes. And then when we looked only at mortality in the third wave, we found that the Delta flag was not associated with mortality. The hazard ratio was 1, but we only had 480 deaths, so it's very small numbers still. And it's quite possible that cases included here would have gone on to die later. And so if we repeat this analysis at a later time period, it might be that there is a difference. What's also interesting to note is that while increasing age was associated with admissions, it was also very, very markedly associated with deaths in this third wave. And then moving on to the reinfections. So we had some data on the recorded reinfections. That's individuals who've had 2 positive COVID-19 tests more than 90 days apart. Obviously, this is just a small proportion of all the reinfections that happened in real life. But it does, we can include it in our analyses. And what we found is that having a recorded reinfection seems to be at least somewhat protective against severe outcomes in this third wave. But again, we had very small numbers. We only had about 50 individuals who had their first infection in the first wave and 150 who had their first infection in the second wave. But just at this point, there is a signal to suggest possibly that previous infection does provide some protection. Next slide. And then in the middle of June, the Department of Health was forced to implement testing restrictions. So this meant that only people who will be admitted to hospital, who had comorbidities or were older than 45, only they could qualify for a PCR test. And so we repeated the same analysis for severe outcomes in the period prior to the testing restrictions and then directly afterwards. And what you can see is that in the period afterwards, the hazard ratio increased slightly to 1.6, but the confidence intervals are still overlapping. But in the period prior to the testing restrictions, the hazard ratio actually decreased to around 1.1. We're still busy teasing out the exact implications of this, but again, here we're dealing with very small numbers because there were very few cases and there was also very little delta around at that time period. Next slide. And then this is apart from the survival analysis, we also just wanted to look very briefly if the younger ages were associated with Delta infection. And this is regardless of disease severity. So what we've done here is just a plain logistic regression for Delta, the Delta flag as the outcome. And we limited ourselves to looking only at the public sector in the period before the testing restrictions were introduced. And then adjusted for all the different sub-districts. And here what we found is that older age was potentially protective. So if you compare the age group 0 to 20, compared to them, those aged 20 to 50 had an odds ratio of 0.77, and those aged more than 50 had an even smaller odds ratio of 0.62. So this does suggest that older age is protective. But we do have to bear in mind the slightly erratic testing patterns of children and young adults. And so what this analysis really can say is that in those who presented for a test, a younger age was more associated with Delta infection. And again, this is regardless of disease severity. And then lastly, in conclusion, so what we found was that this Delta flag was not associated with increased mortality in our third wave at the moment, but it was associated with worse outcomes and particularly hospital admission, even after adjusting for various factors. This is when compared to other circulating variants and the Beta variant in particular. But as there is a high proportion of Delta infections that are mild or asymptomatic, and those individuals don't go for testing, it really complicates the interpretation. And so all that we can confidently say at this point is that amongst those who test for COVID-19, and particularly now with the testing restrictions in place, amongst those individuals, having the Delta flag was associated with a worse outcome. And this is consistent with international findings that Mary Ann presented earlier. And then just before I end up, there are a lot of caveats for this analysis. There's a very preliminary look using this novel proxy for Delta that we're still busy sort of refining and validating. And what we're also trying to tease out is the fact that the seeding assay was used in different geographical areas, and different geographical areas are experiencing larger and smaller third waves depending on previous immunity. So we're just still busy trying to untie all of that complexity. So that's all from my side. Thank you.
Great. Thank you very much to all three speakers, Mary Ann and Marvin, for a very comprehensive overview of those topics, our current epidemiology and the variants, and then to Hannah for sharing that early data to understanding the implications of Delta in our setting. I've got a question for each of the speakers, and then I'll go over to Mark Sundrup, who will address questions that are coming in through the chat. But I'll start off with Mary Ann. And so, Mary Ann, do you have any sense now from having seen the seroprevalence data after wave 1 and after wave 2, what we might expect in terms of what we might be reaching in terms of seroprevalence in the Western Cape after wave 3? Or is it just too early to answer that question?
So, I mean, part of our thinking is it really worth doing seroprevalence again post wave 3, having done it extensively post wave 1 and post wave 2? And can we not just guess what's going to happen? And certainly compared to when we looked at the end of wave 2 compared to at the end of wave 1, we saw pretty much everywhere about a 20% increase in seroprevalence. So no matter how high you were at the end of wave 1, you still went up. Having said that, by the end of wave 2, Khayelitsha was sitting at 70%. And given that they've had a relatively small wave 3, can that really be pushed up to 90% with just another wave? So I think we probably will see if there's a way that we can actually try to look at this, maybe in a more targeted way with not doing quite so many samples. And then the other thing I would just add is our private sector seroprevalence at the end of wave 2 was really very low, around 25%. And obviously we know with Delta being more transmissible, to try to slow down transmission with a higher reproductive number, one would need seroprevalence incredibly high, or a vaccine that prevents infection very effectively with very good coverage to slow that down. So I think we don't know, but I think what we've learned is you've got to go very high in terms of your immunity before you can stop this thing. Yeah.
But I mean, it just the seronegative pool of people that will be affected by a fourth wave, provided there's not a variant that evades immunity, must be getting smaller and smaller in certain communities. And as you've shown for Khayelitsha, but for other communities as well, one would hope.
Yeah, certainly. And unfortunately, I mean, we don't have granular data for all of those suburbs that had a smaller second wave. But I think that's certainly the case. And certainly the people living with HIV are a proxy for that. So I think that is the case. And even outside of Khayelitsha, in the rural areas, West Coast and Central Karoo both had lower seroprevalence post wave 2 and have seen these worse wave 3. So I think that is the case combined with immunization.
Yeah. Okay. Thanks, Marianne. Marvin, I wanted to ask you, just, obviously there must be a lot of researchers around the world looking at the data that's accumulating in terms of the variants and the mutations. And there must be many people looking at predictions as to what mutations might emerge and where the virus might go next in terms of mutation. Any kind of predictions that are out there that give us a sense of what we can expect, or is it just too stochastic and too unpredictable as to where viral mutation might go?
I mean, I'm certainly not a kind of predicting man, so, I have kind of this conversation with kind of Darren Martin, and he's kind of done some work, and in fact there's going to be published soon in Cell. What they do is they kind of monitor on all the kind of virus sequences that they deposit, and looking at particular mutation that is emerging as kind of independently more likely to have kind of amino acid change instead of just a silent mutation. And they have certainly picked up some, and some of which has kind of come to fruition in kind of more recent samples. And then they actually spread across not the virus, not just spike, but also other parts of the virus. So yeah, that is a very interesting one. And not, I can't say that I understand all those predictions, but I think the general principle is still that it is fairly unpredictable, but we know that the one thing the virus is going to try and do is continue to have kind of these immune evasion tactics, and because that's what the kind of selection pressure is kind of requiring answer. But yeah.
Okay, thanks, Marvin. And then Hannah, just in terms of the data that you showed, as you kind of very clearly showed that it's preliminary data, it's an early analysis, but it's fascinating that you see this increase in severe infections when you combine hospitalization and death, but you don't see the impact on death. And any thoughts as to what might explain that?
I think, the one thing is just that a lot of the deaths haven't occurred. So quite a lot of the cases included didn't have enough time to pass away. And obviously there are data delays and things. So at this point in the wave to be making sort of an assessment of mortality is a little bit premature. But we will just have a look at it for now to see where we are.
Okay, no. So clearly follow-up data, some of those hospitalizations might be people who die, and that might then come out as a signal then. Okay, no, thanks to all three speakers again. I'm going to ask Mark Sundrup if there are any questions from the chat.
Graham, I'm going to sort of narrow it down to two broad themes in the interest of time. The first one is maybe at Marvin. I know you don't like predicting, but both are sort of predictive questions. Perhaps while you are predicting, tell us what you think is going to happen on Saturday, but more virologically bent. The question is, based on what we're seeing and the sort of evolution that you described towards other variants, Lambda, Epsilon, etc., is there any way one can predict the potential transmissibility, infectiousness of those variants? Will they cause more disease? And do you think that our memory T-cell response will protect us from those variants as they evolve? That's the first question. So just put that in your memory bank. The second question, Graham, is around vaccines. The one, I'm going to sort of combine it into two issues. The one is perhaps a more generic question, Graham. I don't know if you can answer it. Question has come through whether there's any move or discussion at a national level around healthcare workers who've received J&J getting a booster of maybe with Pfizer or another J&J booster, something that protects us against the Delta variant a bit better. And the second question then around vaccines, maybe to yourself as well, Marvin, is whether the vaccine we have, in that it prevents infection but still allows for transmission, is it inadvertently selecting for more virulent strains? Over to you. Thank you.
Sure. So those questions really speak to the heart of what we don't understand as how is the virus going to achieve what it has already done, and I mean the clear answer is always that if they are able to kind of improve their transmissibility. And so when we first look at some of the mutations and that we've seen in alpha, it was people kind of understood that to be the mutation that enables kind of improved transmission, and therefore that there would be convergent evolution of all the different variant kind of towards those mutation. But that had not necessarily happened. But the virus just kind of found different way of kind of improving its transmissibility. But I think when we're talking about improved transmission, there is potential that it might be linked to potential increase of severity, and if the whole kind of viral load and the efficiency of the viral replication can take place. And that's probably the one that everyone is watching out for, is the mutation that's around the furin cleavage site. So so if you're able to kind of allow that a spike to be cleared and more readily able to gain entry into the cell, you obviously have kind of this early advantage in terms of first getting into a host, and there it also would have some effect in overcoming the early immunity and have some kind of secondary transmission before your kind of B cell memory kick in. But exactly how it plays out, I don't know. It's not inconceivable that the virus will come and find completely a separate path to actually able to do this. I think there was a kind of the question around whether we need a better vaccine. I don't really have answer for that, but in terms of whether the vaccine will select for variants, I think it's no more than a wild type infection will select for variants, because whichever form of infection that elicits antibody, you're going to have those antibody level drop, because that's just what antibody does over time. And in any of that scenario, you're going to get the same effect, which is kind of partially protective level of neutralizing antibodies, and those environment is where variant will be selected. So not necessarily just the vaccine, although I'm sure it could do that. But if we don't vaccinate to have infection in wild type, it probably would just be the same. So I think my answer is still back that this is a bit of a random process, and we don't know how it's going to happen.
Thanks, Marvin. Graham, the question around booster.
Yeah, I'd maybe start off by saying we are going to see some data from the Sesonke trial, where the Johnson & Johnson vaccine was given to almost 500,000 healthcare workers, and some data on breakthrough infections is going to be presented in the next few days. I think the key message, and it's already up on the MRC website, the kind of key take-home message is there have been many breakthrough infections, but the vast majority have been mild, with very few severe infections and hospitalization. So the vaccine, despite this terrible third wave where healthcare workers have been on the front line, it's protected them from severe infections. So, I think that statement must be made over and over again, that the vaccine is working against severe infections and deaths. Not 100%, and very few medical interventions work 100%, but there has been a very substantial reduction. But there is this concern that we are still seeing breakthrough infections, and I think the question will arise from the data, whether there would be benefit from a booster shot, either with a booster from the Johnson & Johnson or an mRNA vaccine like the Pfizer vaccine. And I think it is to an extent a research question. Whether it gets operationalized or it gets done within the context of a research study, we'll have to see. I think that's a discussion that will happen on both a research science level and a policy level. And, you know, we don't know whether giving the boost is definitely going to prevent those breakthrough infections. You know, one would hypothesize that they would lower it further. But, you know, I think that discussion needs to happen once that data is in the public domain. And, you know, just to remember that, you know, in public health, there's always opportunity costs. So if we were to decide to have a program to revaccinate healthcare workers, it would come at the cost of some people being diverted from the national program to giving people their first shot, to giving people a booster shot. So all of those things will need to be considered in any decision. But I think it is going to be a discussion on the table over the next few weeks.
Thanks very much, Graham.
Any other questions, Mark?
There was one last question, maybe just quickly before we close. Whether we have any seroprevalence data from the UK or Europe to determine whether, you know, if herd immunity has actually been achieved, you know, to sort of drop wearing masks, etc., etc. Anything on that front? Perhaps that's a good place to close off with.
Marianne, any thoughts on that question?
There is a large seroprevalence study that has been done in the UK, but I don't think, I think we don't know for sure, and I think it's very difficult to compare with our setting because they also have a large proportion of their population that is immunized, but certainly levels in our communities are substantially higher. But I think the real question is, with more transmissible variants, how high is high enough? And we really haven't seen evidence, except for modeling data, to suggest that it has to be very high. And we haven't seen evidence of substantial protection at the level of a whole population, rather than just a particular community that may reach those very high levels.
Great. Okay, so on that point, just to say thank you very much to all three speakers. Very relevant talks today, you know, that speak to exactly where we find ourselves in the epidemic in the Western Cape. And we will have our next webinar in approximately 1 month's time. And over to you, Mark.
Yes, Graham, thank you. And again, thank you to Project Echo, and we'll see everyone back in a month's time. Be safe and work hard. Bye-bye.